Hello, my name is Celia Greenwood, and as the co-director of the Ludmer Centre for Neuroinformatics and Mental Health, I'm delighted to welcome you today for the event about ongoing research at the Ludmer Centre, a very timely topic. McGill University is on land which has long served as a site of meeting and exchange amongst Indigenous peoples, including the Haudenosaunee and Anishinaabe nations. We acknowledge and thank the diverse Indigenous peoples whose presence marks this territory on which peoples of the world now gather. The Ludmer Centre was founded in 2013 with a generous donation from Mr. Irving Ludmer to bring together researchers working on understanding brain structure and function, the latest techniques in imaging, the latest methods for measuring genetic and epigenetic activity in cells and tissues, and the latest in analytic techniques on how to bring together all these data. The Ludmer Centre includes researchers from McGill University, the Montreal Neurological Institute, the Lady Davis Research Institute at the Jewish General Hospital, and the Douglas Mental Health University Institute. Today's topic is looking at mental health and its relationship to brain function, and how life exposures and stresses relate to mental health. Today's talks will discuss aspects of stress, brain development, and brain function at different stages of life. We will have two short presentations from my colleagues, Dr. Bagot and Dr. Silvera, and at the end there will be a question and answer session. At any point throughout the event, please feel free to submit questions for the speakers by emailing specialevent.med@mcgill.ca. This email address will be displayed on the screen periodically in case you want a reminder. Dr. Patricia Silvera is an associate professor in the Department of Psychiatry at McGill University, and she leads the genomics and epigenetics pillar of the Ludmer Centre. She is an MD with clinical training in pediatrics, as well as a PhD in neuroscience. She specialized as a neonatal follow-up at the Montreal Children's Hospital and was a CIHR-funded postdoctoral fellow between 2007 and 2009 when she started her independent lab. She has over 130 peer-reviewed publications and has trained more than 50 postdoctoral fellows, as well as PhD students and master's students. Patricia joined McGill in 2016 and is a senior chercheur boursier from the Fonds de la Recherche du Québec en Santé, and she will be our second speaker today. Our first speaker is Dr. Rosemary Bagot. She was recruited as assistant professor in the Department of Psychology in 2016 after a PhD in neuroscience at McGill University, followed by a postdoctoral fellowship at the Icahn School of Medicine at Mount Sinai in New York. She has published over 50 highly cited papers examining how stress throughout life leads to lifelong differences in activity in different regions of the brain and to behavioral changes relevant to depression. She has also examined how maternal care can moderate or counteract these effects. Much of her work is undertaken in animal models, such as in mice, and she is an expert in the use of the latest laboratory techniques, such as imaging, that permits viewing brain activity while the animals are actually awake and reacting to stimuli. Her groundbreaking work has been recognized with the Canada Research Chair in Behavioral Neurogenomics. So Rose, please go ahead.
Thanks, Celia. So it's really a pleasure to be here today and to be able to share some of our research with a broader audience and be able to communicate some of the science, hopefully in a way that will be understandable even to the non-expert. And hopefully we can engage in some interesting discussion afterwards about this and some of the connections with other Ludmer Centre work. So my lab studies how stress shapes the brain. So we probably all intuitively know what stress is, but as a scientific concept, it can be really quite hard to pin down. We all encounter stress to differing degrees in our daily lives, and more extreme or prolonged stressful experiences can have lasting impacts on how we relate to our world. There's a huge range of events and circumstances that might be experienced as stressful, and some may be objectively more severe or more life-threatening, but stress really is by its very nature subjective. So control, or really the feeling of lack of control, can be a very important factor in defining stressful experiences. And of course, the pandemic has introduced a host of new and different stresses to all of our lives, as well as amplifying existing stresses in many of our day-to-day lives. Ultimately, all perceived stresses will activate the same physiological stress system to impact our brains as well as our bodies. So this starts with the release of the stress hormone cortisol, which floods our brains and then acts on receptors that are found throughout the brain. So acutely, this stress response is important in helping us to adapt to challenges. But when this response to stress is either really sustained over a long duration of time or especially intense, the same processes that can be adaptive can actually lead to damage. And over time, this can permanently change brain structure and function. For some people, chronic stress will lead to psychiatric disorders, and these may include depression, anxiety, as well as substance use disorders. We know that psychiatric illness impacts the lives of 1 in 3 Canadians, and 1 in 10 Canadians will report having a mood or anxiety disorder specifically. We also know that these kinds of disorders affect more women than men. More than a third of those people affected by psychiatric disorders report that their disorder has meant that they were unable to work at some point. So there's a very profound personal, societal, and economic cost to be factored in to psychiatric illness. So depression can be especially debilitating. For most people who experience depression, it will continue to reoccur throughout their life. Existing therapies, while they work for some people, don't work for many people. And when they do work, they're effectively working by suppressing symptoms rather than treating an underlying cause. We know that a major challenge to developing better treatments is simply that we don't fully understand what causes these illnesses, and we don't know what exactly is going wrong in the brain. And while we know that stress is a major risk factor, we also know that most people who are exposed to stress will not go on to develop a psychiatric disorder. So this observation adds to the puzzle, but we can also see it as an opportunity. We can leverage these individual differences in how individuals respond to stress to understand something about the mechanisms of what makes people susceptible. We can compare the brain function of individuals that respond differently to the same kinds of stressful experiences and understand how their brains are changed. And this can give us a window into understanding mechanisms of various psychiatric illnesses. In my lab, we're using genetically identical mice that respond differently to the same chronic stresses. And then we're studying their brains to find new mechanisms of this differential adaptation to stress. One of the obvious changes that we see in our mice after chronic stress is in their social behavior. So after periods of chronic stress, we see that some mice will avoid interacting with other mice, but other mice will continue to seek out social interaction. And we can use these differences to identify the mice that are susceptible to chronic stress versus those that are resilient. So the susceptible mice are those that avoid interacting, so their behavior has been changed by this period of chronic stress, but the resilient mice are able to adapt and continue seeking out this social interaction. We then study these different groups to understand the neural circuits that are altered in stress-related disorders. And this work can be helpful in determining mechanisms of disorders such as depression. But I think it's always very important to emphasize that depression is fundamentally a human disorder. But nevertheless, we can use tools, we can use animal models to gain insight into mechanisms, basic mechanisms of how stress changes the brain, to understand something about how brains are altered in conditions associated with stress, such as depression. So doing this work, we have learned that stress changes how brain cells talk to each other, shaping how areas of the brain communicate across networks. So we've discovered that stress has different effects in the brains of resilient and susceptible mice. So our resilient mice that continue to seek out social interaction after periods of chronic stress have strong communication between two areas of the brain, the prefrontal cortex, which is important in decision-making and executive control, and the nucleus accumbens, which is a brain region that we know is very important in reward and motivation. Whereas our susceptible mice, the mice that show social avoidance after periods of chronic stress, these mice have a weaker connection between the prefrontal cortex and the nucleus accumbens, but they have a strong connection between the ventral hippocampus, which is an important region for emotion and responding to stress, and the nucleus accumbens. So we've also been able to show, using optogenetic tools to control the activity of specific brain cells in awake behaving animals, that we can make susceptible mice resilient and vice versa by retuning these connections. We've also discovered ways in which stress is permanently shaping gene activity in the brain. So like a dimmer switch, stress can turn on and off groups of genes that control activity of brain cells within these brain regions where we've seen differences in connectivity. RNA sequencing technology gives us a readout of the activity of all genes in the genome. And by profiling brain regions across many animals, we can amass large amounts of data that we can then use to uncover patterns that change with stress. We can also use this approach to identify key regulators that control these interesting patterns of gene activity. So in this way, we identified groups of genes that are differentially turned on and off in the brains of susceptible and resilient mice. We found a group of genes that was turned on in the prefrontal cortex of our resilient mice that keep seeking social interaction. And we then used tools to increase the activity of these specific groups of genes in the prefrontal cortex. And we were able to show that this was enough to make mice resilient to future stress. So ultimately, this kind of insight can help us to identify new drugs to target stress effects in the brain. But also, by looking at how this group of genes is organized, this work has led us to identify a new gene, SDK1, that we know is important in controlling the activity of the wider group of stress-sensitive genes. From here, we're zooming in to understand more about what this gene, SDK1, is doing in the brain. These are pictures taken from the prefrontal cortex of a mouse. Using a technique called RNA-scope, we can visualize individual copies of our gene of interest, SDK1, in brain cells of mice. So in this picture, each red dot is one mRNA copy of SDK1. So we can identify what kinds of brain cells have many copies of SDK1. Where we see green, we know that these are excitatory neurons. And where we see white, we know that these are inhibitory neurons. We can also ask how these cells that we see have many copies of SDK1 communicate with other cells in the brain. So we're currently testing the hypothesis that SDK1 is important in strengthening this connection between the prefrontal cortex and the nucleus accumbens that we know makes mice resilient. So in this lower panel, we labeled all the cells in the prefrontal cortex that connect to the nucleus accumbens in green. So if you see green, we know that that cell that's located in the prefrontal cortex sends a connection to the nucleus accumbens. And we can see by looking at the overlay with our red dots that many of these cells also have a lot of copies of our gene SDK1. So using these approaches, we're learning about the molecules in the brain that are affected by stress to change brain activity and behavior. Ultimately, our goal is to target these molecules to rewrite the effects of stress. And at the same time, we're zooming out to explore how changes in these genes in human populations might influence how individuals respond to stress. In a new collaboration that was actually spurred by the pandemic, Dr. Patricia Silvera and I are discovering how groups of genes organized by STK1 influence how children respond to stress in early life. So this exciting new direction came about through our interaction as Ludmer Centre colleagues. And I think it's a great example of the innovation that can come from supporting diverse teams. And also how sometimes an unfortunate circumstance like the closing of my wet lab can lead to new opportunities for discovery through discovering new collaborations with existing colleagues. So science is really a team endeavor, and I want to acknowledge the team in my lab behind these discoveries. So some of them are pictured here. If you're interested in our work, you can find out more about what we do on our lab website, or you could follow us on Twitter. And I would also like to acknowledge the generous support of the Ludmer Centre, which really makes much of our work possible. Thanks.
Thank you very much, Rose. A really exciting glimpse into some groundbreaking research with some clues for how to possibly improve or develop improved treatments in the future. As I said earlier, the Ludmer Centre brings together researchers who have different approaches and strategies for studying mental health and disease and how they link to brain development and function. And of course, Rose has just highlighted the gene STK1 and how she is now collaborating with Dr. Silvera to understand how this gene may be an important role also in the human setting. So that leads me to introduce now our second speaker, Dr. Patricia Silvera. She studies how early childhood environment and stress can change health and disease across our lives. She poses questions such as, do mental health problems lead to increased susceptibility to disease? Or is it the other way around, where an increased risk of disease influences mental health? Or is the real answer some combination of both of those things? Her research explores genetic risks together with risks associated with exposures, lifestyle, and their effects on health and disease. In 2019, in recognition of her outstanding work on childhood development, Dr. Silvera was invited to become a core member of the USA-based National Scientific Council on the Developing Child. So Patricia, over to you.
Thank you, Celia. Good morning, everyone. I'm very glad to be here to talk to you about the individual differences in response to stress or adversity. As Rose mentioned on her talk, chronic stress is associated with an increased risk for developing psychopathology. It's also important to highlight that the stress response is a systemic response involving different brain regions and also different peripheral organs and systems. For example, the cardiovascular system, metabolism, immune system, in a coordinated action to promote survival and adaptation to this stressful situation. Provided this complexity, it's easy to imagine that people vary in their response to environmental adversity. Some people are more susceptible and some people are more resilient. And we see this even within the same family. Siblings can be very different in their response to adversity for reasons that we're still not completely understood. We know that the genetic background plays a role on these individual differences. Genetic variation define the biological functioning of the person, how the cells and organs work, and also how the organism responds to changes in the environment. Stressful or adverse events happening during the life course of that person will elicit specific responses that can be more or less intense depending on the genetic background. So it's really an interaction between the genes and the environment that defines how the individual responds and adapts to the surrounding situations. Another important factor to consider is the timing in which adversity happens. We saw that chronic stress in adulthood elicits a response, but there are critical periods during development in which adversity will have both this acute effect, but also can leave a persistent mark or impact on the way that the organism will function in the future. The early environment, including prenatal and the initial years, is one of these critical periods. Adolescence is also another important developmental critical period. Then these individual genetic variations interact with the many events happening during the life course of the individual, and via modifications in the way these genes express themselves and coordinate the function of the cell, the systems, and the organs, they modify the risk for diseases in the long term. So the figure here is focused on the brain and the risk for psychopathology, but again, this happens at the systemic level as well. So we can think of a scenario with multiple possibilities, depending on the genes and the environment interaction at each point in time. One event and its underlying biological response happening at the starting point will modify the responsivity to the subsequent event, changing the developmental trajectory and the health disease pattern possibilities. For each event, at each point in time, specific adaptive mechanisms are triggered and modify the phenotype. And this phenotype is the basis for the next response. Therefore, not only the environment changes over time, but the individual responding to it also changes. This scenario explains why some people are more vulnerable to psychopathology and physical disease in response to threats, while others are more resilient. We see this clearly in our data that I'm going to show you right now. We have a birth cohort based on Montreal and Hamilton called the Maternal Adversity, Vulnerability and Neurodevelopment, or MAVEN project. We have been following mothers and children since birth, collecting information on their environment, characterizing how these children grow and develop, and also collecting biological measures. Based on all this information, we are able to construct a score that reflects variations on the level of stress or adversity that these students were exposed to. You see on the figure examples of the different variables that we accounted for, including socioeconomic status, presence of disease in the child or in the mother, family function, exposure to violence, etc. We then attribute points to the presence or absence of these events. And then we have a score that varies from more negative or more stressful, here in red, to more positive or more supportive, here in green. This score is a continuous measure that can be calculated for every participant in a sample, in a cohort. And we have done this in multiple groups of children in different parts of the world. The effect that we see in the different samples look like this. Here we are looking at anxiety problems in children from even at 6 years of age. You see clearly that children raised in the red part of the x-axis, facing increasing levels of adversity, have increased levels of anxiety. On the other hand, children raised on the green part of the x-axis or in more supportive settings have lower levels of anxiety. This effect is very consistent across multiple samples and also several mental health outcomes, and is aligned to the data that Rose was showing us before. Stress leads to increased risk for developing psychopathology in humans as well, and this is an example in children. What we can also note on this plot is the large variability that exists in the response. While several children exposed to adversity have high levels of anxiety, several others that are exposed have normal levels of anxiety comparable to those children that were not exposed. So we see that some children are more vulnerable and some are more resilient. Today in medicine, that's a very large problem because all our programs for prevention, early detection, and interventions are based exclusively on the history of exposure. And as we see here, some of these exposed children would need to receive specialized follow-up and services, but others would not. What my lab does is to create biomarkers that can help us identify who is in fact vulnerable, as you see here in the same example, and who's resilient. Here we created a polygenic marker using functional gene expression data from animal models that can come from studies like the one Rose described to you at the beginning. Through a series of bioinformatic processes and a multi-level integration of multiple sources of data, we identify gene networks that respond to different environmental exposures in specific brain regions, like on this example here. We characterize the gene network from the ventral hippocampus that, as Rose said, is a brain region involved in the stress response. These genes respond to variations in the environment by increasing or decreasing their expression. So they can be called environmentally responsive genes. Then we integrate this functional information to genetic variation that can be measured in humans through genotyping. This allowed us to develop this polygenic biomarker that identifies differences in the responsivity to adverse environments. By doing this, we were able to identify who's more vulnerable when facing adversity, not only in our MAVEN cohort, as I showed you before, but also in the Singaporean Gusto cohort at 6 years of age. The same effects of the risk for psychopathology could also be seen in larger samples at 17 years of age, and even in adulthood in more than 65,000 individuals. Our marker was therefore effective across the different samples, and the interaction holds true for children throughout development into adulthood. So this is one example of a study performed in my lab, but we are involved in other initiatives aimed at identifying biomarkers to characterize vulnerability to the negative effects of stress, including inflammatory markers, neuroendocrine markers, and brain markers as well. So my lab uses this gene bioenvironment interaction approach to investigate child development and a life course perspective for the identification of these individual differences in responsivity to adverse and supportive environments. We do that by using a multi-level integration of models, animal data, clinical, observational, different data modalities like gene expression, gene variants, neuroimaging, behavior, and biomarkers in the context of big data. I'd like to thank my students and staff, collaborators, the Ludmer Centre that allows all this work to be happening, the funding agencies, and you for your attention. Thank you.
Thank you very much, Patricia, for that presentation that really encourages us to think a little bit differently about resilience and risk. As you can see, different research studies and perspectives can provide complementary understanding and also make it clear how complex the challenge is ahead of us. We need to recognize not everyone is the same. And therefore, not only are some people more resilient and more sensitive, but responses to treatments will also vary. The right intervention is needed for each person. And this research, the two presentations that we've just heard, are pointing the way towards these potential improvements in precision medicine for diagnosis, treatment, and prevention. Innovations at the Ludmer Centre are facilitating this work, building computational infrastructure, platforms, open science initiatives, analysis pipelines to help the research of the scientists that are affiliated with the center. and accelerate the development of new ideas and new concepts and new results. The Ludmer Centre, as I said, is supportive of open science initiatives and science communication strategies, and facilitates access to varied audiences, including health practitioners and policymakers. So with that, I would like to thank both speakers again and open the question and answer session. I see that there are a number of questions that are coming in, which is great. To get the ball rolling, I'm going to start off with one question on what is the biological difference between acute and chronic stress and the impact of those two on health? Can we predict who is susceptible or resilient to stress? Now I will let one of you decide who would like to respond.
Yeah, I can talk a little bit about that for sure. So I think that the difference is that we need to keep in mind is that acute stress elicits this series of bodily reactions that are aimed at protecting life at that moment to respond to the threat. So we have, for example, increased heart rate, increased blood flow to the muscles, to the brain. We mobilize energy to increase the availability of glucose to the cells. We increase blood pressure. So all of that is very important at that moment. But while all these actions are essential for survival and for facing the stressful moment, this is the classical fight or flight reaction that we all talk so much about. But these mechanisms are, if they are continuously activated, they can bring damage to many organs and systems. So, for example, a constant mobilization of energy reserves to increase blood glucose, for example, can contribute to hyperglycemia and can, in the long term, contribute for the development of diseases like type 2 diabetes. The same for blood pressure, for example. So there's a point in which the adaptive responses start to bring damage to many systems. And that's when the effects of chronic stress kick in and we have the consequences.
Rose, do you study this in animal models? Do you have short-term and long-term interventions in mice?
Yeah, it's an interesting question. We're really focused on chronic stress, and this is largely because acute and chronic stress have really such different consequences. So often we see at the level of, for instance, cognition, an acute stress may actually have a sort of beneficial effect. It might support cognition, whereas generally we're seeing deleterious effects with chronic stress, although obviously there's a lot of individual variability. Yeah, so it's quite often segregated within the field because the outcomes are so different between an acute stressor and a chronic stressor.
Thank you both. So I have a question from Tara. For an adult who has a very strong stress response, what is the best way to start changing brain architecture and function to respond to stress in a healthier way? Do we have answers for that at this time?
I think we can certainly speculate. So I think a key insight is that we understand that there's sort of different neural connections that may be stronger in individuals who are more susceptible to sort of mounting sort of this like exaggerated stress response versus those who may respond to stress but then be able to come back to baseline. And so one of the key things that I sort of mentioned briefly is this idea about controllability. And we could think of sort of neural correlates of controllability. And there's a lot of research to sort of point to the idea that prefrontal cortex is very important in being able to shut down stress responses, being able to increase this perception of controllability. So you could think about sort of a neural manipulation. Certainly there are ways to manipulate neural activity. There's transcranial magnetic stimulation, there's deep brain stimulation, it's very extreme. But you could also think about engaging in behaviors that we know will recruit these neural circuits. And so this gets into ideas about sort of relaxation and mindfulness, where certain activities may be allowing sort of increased activity in these brain regions. So I think that's an interesting thing to pursue. And certainly even in animals, where we're looking at, can we reshape neural circuits by increasing engagement in tasks where we know these neural circuits are activated?
There's another interesting point related to this, is that we have a very good amount of evidence to suggest that people who are more responsive to adversity or to stress, the same individuals who respond more in terms of having more damage and more risk for disease, they also respond more to supportive environments and to a healthy environment in general. So this types of behaviors that Rose was mentioning, and also others like exercise and healthy behaviors, can probably have a stronger effect for those that are more susceptible, which is which brings hope to us, because at the same time you have more risk, but you also respond more to positive environments.
Thank you.
One more interesting note to just add on that is that there's like a number of meta-analyses that are coming out looking at where in the brain do different kind of interventions act. And so even just this morning, I was sort of flipping through my notifications and noticed another one that is sort of reinforcing this idea that pharmacotherapies sort of hit different targets in the brain than psychotherapies. And so it seems to be there's a pretty good amount of evidence now that sort of traditional antidepressant therapies are targeting the amygdala, which a lot of people think of as like the fear center. It does a lot of other things too, but this is sort of how it's commonly known. Whereas psychotherapies seem to be intervening at the level of the prefrontal cortex. And you can think about sort of, when we talk sort of generally about psychotherapy, there's a lot that comes under that umbrella. But within certain psychotherapies like cognitive behavioral therapy, a lot of what is being sort of processed and learned is about how to sort of reconceptualize your environment, how to alter your responses to situations, how to gain a sense of controllability. So it's really interesting to see that these different kinds of interventions are changing the brain in different ways.
We have a question from Carolyn. Carolyn asks, what are the potential pharmacological implications of STK1 genetic research in terms of the development of new therapeutics for clinical treatment of anxiety and depression? Would therapeutics target the dopamine pathway?
That's a really interesting question, Karen. So we're actually not looking primarily at dopaminergic pathways. We think that STK1 is involved in rewiring of connections. At the moment, we're studying the prefrontal cortex, and they're primarily glutamatergic neurons, although we think it might also be in GABAergic neurons, so excitatory and inhibitory. But kind of zooming out to sort of what are the pharmacological implications. This relates to kind of the approaches that Patricia's talking about as well, where we kind of zoom out from the individual gene and look at its broader network that it's implicated in. And so STK1 is an interesting target because we hit upon it from this approach of looking at how the relationship amongst groups of genes has changed and identifying whole networks of genes rather than an individual gene. We're then zooming in to find out more about that one gene because we know it's important in controlling that network. But when we have this sort of group of genes that we know is important, there are a number of sort of bioinformatic tricks we can do that can give us hints about ways to target the sort of pathology pharmacologically. So we can take our list of genes that we know is important in sort of that stress sensitivity, and we can compare it to lists of genes. There's these big databases available of lists of genes that are turned on or off by a whole range of different drugs. Some of them might be drugs that are already in use for different things. Some of them are just small molecules that someone made in a lab sometime and then put on some cultured cells. And so we can run these big scans that then generate predictions of this molecule should have this effect on that group of genes. And so that's sort of an early step in the pathway that we can then take our hits from those kind of bioinformatics scans and run them through animal models to see, can we confirm this? Can we potentially identify novel antidepressants this way by repurposing existing molecules?
Thank you. So I have a question from Lilia for Patricia. What do you think makes women more vulnerable to mental health disorders? Are there environmental causes there?
Yes, certainly environmental causes, but also genetic causes. And yes, I think that women have increased prevalence, as Rose was saying. But I like to think about that as men and women being different. And what I mean by that is that the pattern of disease from men and women is different. It's not necessarily that women are more vulnerable only. It's just that the phenotypic presentation of the disease is different. And it's very important to be aware of these sex differences for the sake of diagnosis, for the sake of prevention, for the sake of treatment. And I think that we're making good progress in understanding these biological differences that are genetic, but certainly also environmental and social, and there's a lot involved on that.
Thank you. I have a question here from Bernard from Berlin, who asks, how is your research going to impact and guide treatment of patients with stress anxiety in order to make them more resilient? I think these are things you've touched on in your presentations, but we'll probably come back to them a little bit.
Do you want to say something, Rose? Yeah.
Maybe just sort of like as a general comment, I think that one of the sort of big themes that comes through with both my work and Patricia's work is the idea that it's not just what we see after stress that determines these differences. And so I think often we look after the fact and then we try to understand what's going wrong. But I think what our work points to is that there's something that's already different beforehand, and that if we can understand what that is, but even at a very low level, if we can just identify the individuals who are vulnerable, that gives us an enormous opportunity for intervention, because we can intervene at a time before the brain has already been altered, before the body has also been altered, because obviously there's this brain-body connection. So I think that's sort of like a big sort of overarching implication of this work.
Yeah, and I think that the place where we want to go in the future is to have more tailored approaches to medicine. So as we identify these individual differences and we know the difference that happened beforehand, as Rose was saying, we can have a better, a more personalized way to again, diagnosis and treatment. But there are other implications of our work as well that go beyond, molecules and pharmacological agents. So one example to talk about this happened last month here in Quebec. So we had a law that was approved, Law 83, and it's been more than 20 years that many entities have, like the Médecins du Monde Canada and partners like McGill and the International Bureau for Children's Rights, and all these entities have been asking the government to extend the access, the health access to RAMQ services for children born from parents in vulnerable migratory status, like international students, for example, temporary workers. And this entity, Médecins du Monde, reached out to our lab a number of years ago, and they started to use the cumulative adversity score that I showed you here in the talk. So they use this information on their action. So this is an example of how the work done in the lab can contribute to help make a difference in the lives of children and families. So I think, of course, identifying molecules and pharmacological agents is very important, and that's where we want to go, but there's a broader impact as well.
Thank you. I have a question here from Michael. It's a bit of a follow-up, I think, to your previous response, Rose. Is there a link between the conscious reaction to stress and the physiological and damaging effects of stress on the body? Does the perception and feeling of being stressed affect the physiological effects of stress? And you had made a comment about a brain-body connection, so maybe you could elaborate a little on that.
Yeah, so certainly. I think the first thing to comment on is that, so to activate the, for like a psychological stressor to activate the physiological stress response, it has to be perceived as stressful. So there's a lot of room in there for this sort of like conscious perception of our experience to shape how we respond to stress. But then I think you can also imagine that you then have potentially sort of like cyclical effects of like, once that stress response is launched, that's going to feed back onto how you perceive your ongoing environment and sort of circumstances around you that could continue to amplify versus maybe sort of shutting it down.
Thank you. I don't know, Patricia, do you want to add anything, or?
Yeah, I think that the notion of perceived stress is very important, that Rose was saying, is that, and she emphasized this during her talk, that of course we know that there are different intensities of stress, but it is a subjective experience, and the perception of threat can elicit the same response, which is very interesting, and people vary on that as well, right? So there's a lot to account for, I guess, when we are trying to identify who's vulnerable and who's resilient.
You talked a little bit about we had a question on males versus females, women versus men. What about age? Does chronic stress impact people of different ages differently? Yeah,
that's a very very good question, and we touched on this a little bit during the talk, and yes, definitely. So we know these effects of chronic stress in adulthood, as we discussed here, but when happening during this periods of development in which the brain, for example, is growing and making connections and establishing its functioning, adversity or stress can leave persistent marks on the way these connections are made, or the way the neurons talk to each other and synapses are formed. So it changes the way the individual responds persistently over the life course. So the response itself also is different, like the markers of stress responsivity can be different in young kids versus older kids versus adults. But I think that what's the important message here is to understand that events happening early in life can leave a mark and modify the way the individual will respond in the future. And this can contribute for the increased risk for disease later in life.
And then I think thinking about sort of the other end of life, there's sort of a different trajectory of changes with cognitive aging and sort of healthy as well as pathological cognitive decline. And I think this is an area that really needs a lot more study about the interaction of stress, and particularly chronic stresses, and even things that are maybe less obvious as chronic stresses, such as sort of social isolation. But we know from animal work and human work, this has profound effects that this is going to be interacting with the changes that are happening in the brain with aging.
Yes, absolutely. And I think this leads really nicely into a question from Laurel from South Africa. I'm not sure that we have answers here, but she's asking if you think sort of globally in terms of social and political upheaval in large populations around the world. It could be COVID-19, but it could be violence and food insecurity and many other things. Do you have any suggestions for population-based approaches for prevention and intervention? I don't know. Yeah, Patricia, if you'd like to start.
Yeah, I don't think I have like a definite answer on that, but at this point, I think one important action is awareness of these effects. And this is very present in our lives. Like here at the center, we're always talking about stress and chronic stress, but it's not true for everyone and for every place in the world taking care of these issues. So I think there's a lot that needs to be done, starting with awareness, but also other actions like promoting access to healthcare and to interventions and to support in the communities. It's difficult to imagine a global response. Probably the answer would come from smaller groups attending to the needs of that community, right?
Yes, and I'll add myself that awareness at the political level, of course, is a really good place to start. And we're so happy that you are all in attendance here today as part of that awareness. There's a bit of a follow-up question on this topic that I've received from Brian. Is there a way to detect that there might be somebody who's particularly susceptible at an early age?
Yeah, I think that that's the work we were doing, and we have been working with other partners as well, trying to exactly create a set of markers that can help us identify who's vulnerable. Especially for young kids, this is not, it's not very well studied. So there's a lot that needs to be found out about that. But up to now, as I said before, like most of our interventions and diagnosis and establishment of therapeutic proposals are based only on the exposure. So we do need to improve the way we identify these responses, not only in childhood, but also in other ages. So
we're getting close to the end of the hour, not quite there. I'd perhaps like to close out the question and answer session with a crystal ball. Start by asking Rose, what do you think is on the horizon in this field in terms of upcoming research results and findings?
Yeah, that's a fantastic question. And it's a really exciting time to be a basic neuroscientist, because we just have this incredibly rapid evolution of our toolbox. And we can do things that I never imagined would be possible. So some of the things that we can do, we can essentially like put a microscope in the brain of a mouse while it's awake and behaving, engaging in a complex behavior, and we can record activity from brain cells. But beyond that, we can know exactly what brain cells they are. We can know what other cells they connect to. We can know what their genetic profile is. And I think this gives us enormous power to test very specific hypotheses. But what I would say is that that power will only really be realized through these kind of collaborative and translational endeavors where we move back and forth from the basic research and the human research. And I think that this is something that's starting to be appreciated, but it's still undercapitalized, and it's still difficult work to do very translational work. In a large part because sort of funding mechanisms take time to catch up. And so I think that is what I would see as the big thing of the future, where we develop a hypothesis based on sort of a human observation, and then we use our tools as basic neuroscientists to get really precise insight on that.
Thank you. Patricia, can I ask you the same question? What do you see on the horizon?
Yeah, I agree with Rose, and I think that we are lucky here in the Ludmer Centre because we're very close to each other, and then this translation can happen among ourselves and other collaborators that we have around here. So I agree, like integration of these different types of data, going from the very specific and essential work that is done at the bench, like the one that Rose showed you, and then using this information to identify if the same mechanisms are existent or are true for humans, are important for human disease. I think that's the next step, and we're very close to getting there. So I think it's promising.
Thank you both. A last comment before I wrap things up from either of you, or should I go ahead and wrap up? Okay. All right, so I would like to express a very warm thanks to the audience who for their interest, for attending today, and for all the questions that were submitted. I'm sorry we didn't manage to get to all the questions. If you have something that's a really burning question, there would be a possibility of following up by email afterwards. On behalf of the Ludmer Centre and on behalf of McGill University, we're very honored to have Dr. Baggott and Dr. Silvera among us today, and for their really wonderful presentations and answers to questions. Thank you so much to you both for your clear and presentations and your insights and all of the ideas for the future.
Thank you.
Thanks.
Before closing, I just want to make a note that McGill officially kicked off its bicentennial in March of this year, and there will be celebrations over the year, including the launch of the 3rd century, alumni events and online activities. You can stay up to date by checking out the McGill alumni website at alumni.mcgill.ca and the official university bicentennial website, which is 200.mcgill.ca. So thank you again to everybody who attended today, and I hope you enjoy the rest of your day. Goodbye.